So now I'll invite Magnus von Knebel Doeberitz to speak about virus-host interaction at the transformation zone. Magnus.
Thanks a lot. Yeah, what I would like to do is to focus a bit on aspects that relate the anatomical features of HPV infections back to the molecular basics that might be the reason why we observe these clinical pictures that have been discussed in the morning. So as you have heard in the previous sessions and previous talks, papillomavirus are almost everywhere. You can find them in the genital tract, you can find them in the oropharyngeal tract, you can find them beneath your fingernails and wherever else. But the problem is that we usually don't know which kind of cells are actually infected by these lesions. What we can observe in a clinic is a very particular feature that is common to all papillomavirus. They either don't cause any clinical signs, as most of the papillomavirus you have just heard by John Doba, or there are others that cause lesions that usually grow out as clonal lesions. So taking just a normal wart, there's usually one wart that grows on a certain site. This wart is full of virus that is released at the top of the surface of this wart, is spread in the surrounding epithelium, and most likely there are many little scars or lesions that allow these viruses that are released at the surface of these lesions access to the basal membrane and potentially could initiate another infection cycle. Now, the observation clearly is that this is a pretty rare event. So through the evolution of papillomaviruses in man, obviously we all have developed very tight mechanisms that protect us from the pathogenic effects of these viruses. We may get a wart once upon a time. This wart usually regresses and we forget about the disease. But what we actually don't know is what happens to the virus and the cells that do not show any pathological features. Are there cells that become infected by the virus and that do not grow out to any further lesions? Totally unknown. And if we anticipate that there is the fact that papillomaviruses infect other epithelial cells, what actually happens to the virus? Why doesn't the virus become active in the very same cells where it usually causes disease in other circumstances? Now, John has already shown you this phenotype of the tree man, and so far totally uncharacterized disease. Obviously genetic disease because it occurs clustered in certain families. However, what the basis of this disease is not clear so far. But we can anticipate that these guys have something or lack something that most of the other individuals have, a very tight genetic mechanism that controls HPV infections once those viruses can enter a cell. Now these lesions are due to HPV infection, HPV2, the classical skin type, but there might be some other conditions that we have not yet realized also for other papillomaviruses. Now, taken together, then we can, coming back to the cervical lesions, there's another observation. All these cervical lesions that you observe in the clinic and that are of clinical importance arise in a very tiny epithelial area around the transformation zone of the cervical epithelium. There are very few lesions that occur on the outer sides of the lesions. There are few in the vagina or the vulva they occur, but they are usually somehow related to other aspects, and most of the vast majority, particularly of the pre-malignant lesions, clearly occur or arise from very few cells that are just clustered here at the transformation zone. Now this is something peculiar, and we can then think back what actually makes these cells so sensitive for HPV transformation, or the other way around, what protects all the other cells so very vigorously against the pathogenic effects, even the oncogenic effects of human papillomaviruses, although they presumably are being infected by the very same viruses. Now, if you then think this further and consider all the other lesions of major clinical importance, then in the anal tract, for example, particularly in homosexual men, you observe the very same phenotype. The lesions, the preneoplastic lesions that are induced by HPV are usually classical intraanal canal lesions that are arising at the transformation zone in the linea dentata in the very same localization in comparison to the transformation zone at the cervix. And again, we have heard already this morning from Laura Gillison's talk that the very similar phenotype occurs in the oropharyngeal cancers that are related to HPV. They usually all occur in a region where you have the Waldeyer epithelium, that is a very tiny epithelium that covers the lymphoepithelium among the tonsils and the other immunologically relevant organs in this area. Now, something obviously that is very specific to few cells that are more sensitive if it comes to transformation or oncogene activation than in most of the other tissues that could potentially be involved. Now, considering these aspects, we have to realize that there are many totally unsolved questions still in the HPV biology. First of all, we don't have any idea how many epithelial cells actually become infected because we can't measure it. If the virus doesn't do anything to these cells, we don't observe it, and then we don't measure it. How many of these infected cells actually start replicating the virus, giving tiny, very tiny little lesions that are clinically hard to observe, not known. We know that men are infected at the same ratio as women, but how many lesions are actually occurring in men is characterized so far not in great detail. How many of these HPV-infected cells start to express the E67 oncogenes in a deregulated manner, as John has just explained to you, initiating actually a neoplastic transformation process that then triggers chromosomal instability, also not known so far. How many of those, how and why does this happen in what kind of cells? Are it only the cells at the transformation zone? Presumably yes. And if you consider this, what is actually the reason why it particularly happens in these particular cells? What is different in these cells compared to all the other squamous epithelial cells that are becoming infected by the very same oncogenic HPV types? Now, let us take a bit closer look to the transformation zone. The transformation zone is considered as an anatomical area where you have cells that can either differentiate in squamous epithelium, or if you add a bit more estrogen, the very same cells can differentiate into some kind of glandular cells, the columnar epithelium. Now, the very same cells are hormone sensitive, much more hormone sensitive than the cells that are a bit apart from this region. So we can speculate that this is a certain region where very specific cells that are different from either the glandular cells or the squamous epithelium has developed. Now, the landmarking paper of Christopher Crumb's group, Michael Hurst, was published this year in PNAS. And what they did was to characterize the gene expression profile of these cells at the squamous columnar junction. And what they clearly could show is that the gene expression profile in these few cells located here at the squamous columnar junction is quite different from what you can observe in the squamous part of the epithelium, or of course of the glandular part of this epithelium. So the biology of these cells, at least based on the gene expression profile, is something different from what happens in a normal squamous cell epithelium or the glandular cells, suggesting that there is some secrets behind that allow us to, or that are important for the biology of these HPV infections. Now, to summarize all what we know now in a little scheme, we know that there is obviously access to the basal membrane required for an HPV infection. We can anticipate that these infections take very often, or happen very often in a natural environment. And we don't know why some of these HPV-infected cells obviously start to produce a clinically relevant lesion. What we can observe is that these lesions are usually clonal in their clinical growth pattern. So they produce virus, as John has just explained to you, in a very tightly regulated way, depending on the differentiation status of these cells, clearly suggesting that the expression of the viral genes is very well orchestrated with the expression of the individual cells throughout the maturation process of this epithelium. Now, if you consider HPV-16, this happens in men, this happens in females, that happens in the genital tract, and presumably in many other sites of the body. However, what is particular to females, again, is the one event that happens obviously in the squamous columnar junction area, where these particular cells allow the virus to deregulate its oncogenes, the 67 oncogenes, that all of a sudden in some few of these cells start to become overexpressed. This can nicely be highlighted by staining those lesions for P16, an indirect marker of the oncogene E7 activity. And these lesions, these transforming lesions, are predominantly observed, particularly in these squamous columnar junction epithelium. Now, what is the basic mechanism that then governs these phenotypes, and what happens to the HPV genome in these settings? Now, we have seen that the HPV life cycle is clearly dependent on the squamous epithelial differentiation and maturation. That's forgiven. The tissue differentiation in many instances, and this has been shown for virtually all other organs than the skin, depends on epigenetic mechanisms that control these, the genes within these cells. So there are certain methyl groups that can bind to the DNA and tell a gene, you need to be expressed under these circumstances in my life cycle of the cell. And there are other mechanisms that can take off these methyl groups and can activate the genes usually. So epigenetic modification is a very flexible biological system that allows certain genes to be expressed under the one condition and allows the very same gene not to be expressed and alters expressions. Many hormones are working like this, cytokines are working like this, so they simply activate genes by taking off methyl groups or inactivating genes or larger groups of genes by simply adding these type of methyl groups. So then we can speculate that this very same milieu, which obviously governs the regulation of the cellular genes throughout the maturation process of the epithelium, might have also an impact on the papillomavirus genomes, explaining these peculiar gene expression profiles in the various stages of squamous epithelial differentiation. So having said this, we speculated that particularly these genes of the virus that are quite important for regulating its own expression might be the primary targets of differential epigenetic modification in the upstream regulatory region of the virus. That is actually the promoter and enhancer element that is located in the upstream regulatory region. And first of all, the papillomavirus E2 protein comes into mind because that's the master regulator of gene expression, at least in the early stages of the papillomavirus life cycle. Now, the papillomavirus E2 protein binds to various sites that are evolutionarily highly conserved throughout papillomavirus evolution, throughout all the various types of papillomaviruses. And there are actually, at least in the oncogenic HPV types, 4 binding sites for these papillomavirus E2 protein present within the promoter element. There's one which is referred to as the papillomavirus binding site 1. In some references, it's also called the E2 papillomavirus binding site 4. Don't get mixed up there. And if E2 binds with a very high affinity to that particular site, it slightly activates the promoter element, allowing for a little bit of gene expression in the very early phases of an HPV infection. Now, this mechanism continues, so the virus starts to produce, under this influence of under expression of the E2, a little bit of early genes. The early genes produce more E2, and the more E2 is produced, the more active this mechanism comes, until there's enough E2 in the very same cell that then binds to low affinity binding sites that are more downstream of this E2 binding site 1, particularly in the promoter region, particularly the E2 binding site 4. If the E2 binds here, it competes with the Tata box binding factors and then suppresses thereby the papillomavirus gene expression profile. So using this very simple mechanism, the virus has invented a mechanism that keeps its gene expression on a very low level in a highly orchestrated equilibrium in the basal cells. And this is the reason why the virus just produces as little E6 and E7 in the early stages of the infection that it needs in the basal cells that are still replication competent, but then can shut off the very same expression and downregulate the very same promoter element in the same element. So a very tricky system, and details have been reviewed in a very nice article by Hedge et al. 2002. So now, if we consider this, then we can think about that these mechanisms might be an ideal target for epigenetic regulation. And if you look back then in the gene bank system, you easily realize that the few CpG elements that are targets of differential methylation in genes are actually located particularly in these E2 binding sites within the viral genome. So there's a cluster of CpG denucleotides that are evolutionarily conserved within these E2 binding sites, but not elsewhere, at least in the regulatory region of the papillomavirus. And this tells something, suggesting that these epigenetic modifications that take part in these sequence elements might really be of functional relevance. So thinking about that, we went back to the literature and reviewed all the papers that have been published that suggest that indeed the papillomavirus genome is somehow methylated throughout the life cycle. A very early report published by Frank Rösel showed that differential methylation indeed has an impact on the gene expression profile in cervical cancer cells at that time. There are many papers that report that methylation of the E2 binding sites impact E2 binding and modifies thereby the promoter activity. HPV becomes increasingly methylated even in other sites of the virus with the progression through the or pre-neoplastic progression on its way into invasive carcinomas, suggesting that this epigenetic regulation might be used by the virus to downregulate other genes that are not required for the neoplastic progression. And finally, there are a couple of papers that showed that using hypermethylation of these genomes could be even used as a diagnostic marker, a very decent diagnostic marker in other settings. Now we then continued thinking about what happens to the biology of these lesions, and the first question we had was, can we actually identify papillomavirus genomes in simply normal-looking squamous epithelium that doesn't show any cytopathogenic effects? Thereby we used epithelium that was located directly adjacent to an CN1 lesion. On the very same aspect, we asked whether we can identify other methylation patterns, epigenetic methylation patterns, in productive lesions, where the virus really goes through all its phases of the life cycle, particularly in CN1 lesions. And the final stage then was to identify the very same methylation patterns in lesions that already underwent this transforming mechanism, where the E67 genes have been activated, and this was indicated in these experiments by P16 overexpression. And by doing so, we microdissected with a laser beam the various parts of the epithelium. So we microdissected first the basal layer of this epithelium, then we cut out the superficial part of the epithelium, and finally isolated the intermediate part of the epithelium and extracted DNA from these various parts independently. The very same approach was used for the CN1 lesions and also for the CN3 lesions and the transforming epithelium. Now, to summarize a lot of data in a very brief overview slide, what we observed actually was that in the normal tissue, where the HPV16 genome was clearly present, all the CpG islands that were present in the viral genome were heavily methylated, suggesting that that most of our cells actually possess a protection system that immediately, if the virus enters these cells, puts a lot of methyl groups on the viral genome, thereby preventing its genetic activity and makes the virus as a silent bystander that doesn't do any harm to the various cells, explaining a lot of the non-disease part of this clinical story. Now, in the productive lesions, there was a very complex pattern that I don't have time to go through in detail, but remarkably, the early promoter, particularly the E2 binding sites 2, 3, and 4, were heavily methylated in the very superficial cells, suggesting that in this setting, the methylation of these binding sites simply shut off the early promoter, allow for gene expression of the late genes, the L1 genes, that are not represented on these slides here. Now, if it comes to transformation, the few cells that are actually sensitive to to obviously differential gene regulation in the squamocolumnar junction, then we observed a tight methylation, particularly of the E2 binding site 1, and in some instances also the E2 binding sites 3 and 4. Now, methylation of the E2 binding site 1, we were very surprised because we thought if the E2 binding to that particular site is blocked by methylation, then the viral genome should be shut off. Nothing should happen. But what happened actually was that if you use this in experimental systems and particularly methylated just this one site here, the promoter didn't become inactivated. There was another transcription factor complex recruited to this particular site, and this superactivated this particular arrangement of the site. So this is a wonderful example that methylation of this particular CpG nucleotide within the E2 binding site becomes superactive upon methylation instead of being downregulated. Now, what's about the other sites? If you then consider this as a kind of a model, we realized that E2 binding leads to some promoter activation. If E2 binds here to the downstream sites, it comes to promoter inhibition. So this is the model of equilibrium. Now, in larger, more extensive studies, we also identified more heavy methylation of the E2 binding sites 3 and 4 in the promoter region, along with this methylation in the E2 binding site 1. How can we consider this then in a model? Our current model and hypothesis is that E2 binds to the methylated E2 binding site 1, and this leads to superactivation of the promoter. Now, if then the downstream E2 binding sites are also methylated, this doesn't allow for E2 binding, simply preventing inhibition of these promoter sites by adding methyl groups. And the consequence of this is a superactivation of the complete upstream regulatory region. So we can summarize this model that site-specific methylation of CpG nucleotides, obviously within the upstream regulatory region, appears to be responsible for the differentiation-restricted gene expression profile, as I've just shown you in the normal life cycle, going from the basal intermediate to the superficial cells, and then to the aberrant methylation in the E2 binding sites 1, 3, and 4 that gives or allows the superactivation of the 67 oncogenes that then finally transform the cells into chromosomal instability and all the consequences that has in pre-neoplastic progression. Now, we therefore can conclude epithelial cells at the squamous columnar junction obviously retain a mechanism that allows for this particular sequence-specific methylation that gives rise to the methylation in the sites that I have just shown you. What that mechanism is, we don't know, but we hopefully will clarify in the next few months. And the work that I presented to you has primarily been done by Svetlana Vinokurova, Elena Prigge, who is doing this in the oropharyngeal epithelium, and Miriam Rauschenbach and some other people. So we were continuously supported by pathologists, Dietmar Schmidt and Christine Bergeron. Thank you very much for your